Hi, I would like to welcome you to our CCG webinar series. Thank you for joining us today. The presenter for today's webinar is Sarah Wawitsky, application scientist. And just to introduce myself, my name is Valérie Campagna-Slater, scientific support manager at CCG, and I will be moderating today's webinar. During the webinar, if you have any questions, please use the Q&A window to type in your questions. You can open the Q&A window by clicking on the Q&A option located in the menu below your Zoom screen. Only myself and the speaker will be able to see your questions. We will answer as many questions as we can at the end of the webinar. However, if we're unable to answer all the questions, we will get back to you subsequently. Also, please know that access to recording of this webinar will also be made available within the next 7 days. We'll send an email to everyone who participated to let them know once the video is available. So without any further delay, I'm delighted to introduce you to Sarah, who will be presenting our webinar. Sarah?
Thank you very much, Val. My sound goes through. All right.
Yes,
So
perfect.
thank you, Val. Hi, everyone. Thank you for joining our webinar today. We will be talking about structure-based molecular transformations. Now, the agenda I've written here on this slide is first we'll talk about what are molecular transformations. Then I'll talk about how we've chosen to implement that in Mo. And I'll finish up with a case study where we look at transforming sildenafil sitting in a protein pocket of the PDE5 protein. All right, so first of all, let's talk about this concept of molecular transformations, or medchem transformations, as we call them. The idea is that we can introduce very focused changes to a chemical structure. This will create different analogs that are very similar, except for these very local changes to some of the fragments. This transformation is in general interesting because overall it will leave the properties of that compound similar. So that is the affinity, for instance, but also overall different phys-chem properties will be very similar. So I've shown you a couple of examples of known medchem transformations on drugs. Up at the top of the slide, you see two different PDE5 inhibitors. In the left, we have Pfizer's sildenafil or Viagra, and on the right, we have Bayer's vardenafil or Levitra. Now, those two compounds are very, very similar. There are actually only two differences between them. The first one is that the piperazine ring on sildenafil only has a methyl group, whereas it has an ethyl group on vardenafil. This is an example of a transformation that we call a homologation. I'll come back to that one later. The other thing that has changed is in the scaffold. So this fused ring system that we have both in sildenafil and in vardenafil. In sildenafil, we have the two nitrogens next to each other, so it's a pyrasyl ring. Whereas in vardenafil, we have what we call an NC switch. So a nitrogen and a carbon has changed places, and we have an imidazole instead. So very similar, but nonetheless, they are different chemical entities, but they still have same kind of binding affinity to the PDE5. They also have same ADME properties, same half-life, and so on. Now, in the middle of the slide, you see two different antibacterial agents. First, we have ciprofloxacin, and then we have levofloxacin. Again, the scaffold is exactly the same. The only difference is that we have an epoxy ring in ciprofloxacin, which has been cyclized in levofloxacin. And we have a piperazine ring again. It is not alkylated in ciprofloxacin, but we have an N-methylation on levofloxacin. And finally, down at the bottom of the slide, we have two EGFR inhibitors. We have gefitinib or Iressa from AstraZeneca, and then we have erlotinib or Tarceva from Roche. They have the exact same core. It's an aniline kinazolin core. The only changes are a couple of updates out in the R group. So there is a couple of similar groups added to the tails. And also the substitution on the phenyl ring is changed from 2 halogens to an acetylene group in erlotinib. So again, multiple examples of compounds that are chemically very similar and their properties are very similar chemically. They are also similar, but there are these minute changes that make them novel compounds. So when we set out, we wanted to create an application that could introduce exactly these kind of small localized modifications while addressing common issues that we are aware of around molecular transformations. So how did we go about making an application that does molecular transformations? Well, first of all, you need a set of rules, right? And you need a set of rules that can transform the molecule. We're going to look at each of these. So we have something called bioisoters, analogs, heterocycle substitution, homologations, and so on. Now, these rules are, if you're an experienced medicinal chemist, you probably know many of them, or maybe your colleague is an experienced medicinal chemist who knows many of them. You can also find them in literature or in different informatics analysis, but we really want to make them available to everyone. Now, when we do the transformation, we can have two different objectives in mind. Sometimes we want to do something called bioisosters. A bioisostar is a transformation that preserves a property. That could be affinity, it could also be any kind of other property. But sometimes we also want to do an optimization where a bioisostar will remain, and optimization will improve a property. You could even imagine that you had a project where you wanted a combination of those. You might have a compound where you have a good affinity, so you want to create a compound that has the same affinity, so it's a bioisoster in terms of affinity, but you might want to optimize some of the ADME properties, maybe a better bioavailability or a better half-life or something like that. That would be an optimization on the properties there. Now, there are certainly other programs out there that does it. It's definitely an area that many people are interested in. However, many of them suffer from the same kind of issues. So first of all, if you think back to that sildenafil and vardenafil example I showed you in the first slide, we talked about that there was an NC switch in the ring from the pyrazole to the imidazole. Now, I can easily sketch that down. The computer can also easily make it. But can you actually easily create that in the laboratory? Or said in other words, are the compounds we come up with actually synthetically feasible? Another common issue is that the transformation rules lack a context, so they don't have any awareness of the protein. So you could imagine you can create loads and loads of compounds in 2D that are, similar analogs to your starting point, but they may not even fit in your binding pocket anymore, nonetheless even have the same binding mode, so say the same hydrogen bonds. So it's not necessarily valuable just to create loads and loads of compounds if they, first of all, can't be synthesized, and second of all, they don't have the binding mode that we're interested in. And I guess third of all, we don't really want to sit and look through thousands of compounds. So we want to address these issues by introducing the context of the receptor that will help us filter down, score, and rank the output so that we go from potentially thousands of compounds to a smaller subset of interesting compounds. So our objective was to create a structure-based molecular transformation. So we had a couple of things we wanted to do. We wanted to be able to create follow-on compounds, so you can also call them lead optimizations. We also thought about helping the medicinal chemists by filling out their SAR table. And for those of you not familiar with the SAR table, it is, imagine you have your scaffold with different R groups on, and you can have combinations of R1 group versus R2. So have a table of that and different combinations of them. And you'll probably create some of those combinations synthetically, and you will go and test them. But often a SAR table is really sparsely populated because it takes a lot of time and effort to create all of these different combinations and test them. So what we can help out with in the computer is to generate all these combinations and give them a predicted score as well. And when we do these predictions and lead optimizations, we want to do it in a way that simulate the medicinal chemist approach. So we only want to use rules that focus on small scale modifications, or said in another word, local exploration. We want to avoid any kind of gross changes. So for instance, changes to the scaffold, because basically we are not at the stage where we generate new leads, rather we want to optimize what we have. And at the end, of course, also, we want to be able to rank these potential molecules for the MedChem group to pick out the ones that look the most interesting. So our solution that we created was that we want to perform these MedChem transformations in the receptor pocket. We want the receptor pocket to give us context to our rules. We want to be able to create molecules that not only fit in the pocket, but also have a similar binding mode and have similar interactions that we know are important for ligands that bind in that specific pocket. And then we can score them with our docking scores to get a predicted affinity. And we also want to assess their synthetic feasibility by using our descriptor that we call rsynth. So all in all, we want to do a structure-based approach to generate more relevant structures. Now, let's talk about these different localized transformations that we're interested in. So the first one I mentioned was a bioisoteric transformation. You can think of it as a replacement of functional groups. So if you look in the top right corner, you have an amide, and then beyond the arrow, you have a vinyl fluoro. So those two groups are bioisosteres. You can replace one with the other, and they are very similar. They have similar number of atoms, similar size, similar shape, similar polarity, electrostatic properties, and so on. Now, what they also have is something we call similar exit vectors. An exit vector is that bond that connects the fragment to the rest of the molecule. And we are interested in having similar exit vectors between our replacements because that means that the rest of the molecule can stay in the same 3D space, right? We just take one group, take that out, replace it with another group, and it fits in the exact same way. Below the first transformation, you see we have amide to azo. That's also a bioisoster. If you go a bit further down, the fourth one, I always find that interesting. That is a carboxylate to a tetrazole. They are bioisosterics. They don't actually look like it, but they have the same size, the same charge, the same polarity, and so on. Now, we can also look at what we call quasi-bioisosteres. So those would be minor modifications to the properties. They're very similar, but not exactly the same. For instance, ketone esters, amides will fall into this group of quasi-bioisosteres. Another version of that is when we have a spatial extension. So imagine you have a phenyl ring, you can replace that with an indole ring. Again, they're very similar, but they are slightly different. So for instance, the indole is a bit bigger, and also the way it connects out these bonds to the rest of the molecule, they spatially sit slightly different. Now, when we create rules, we have to keep in mind that sometimes we want to restrict some of these rules a bit. So for instance, you might want to avoid halogenation. So say you have a hydroxyl group and you can replace that with one halogen or the other halogen or the third halogen. Suddenly you get a huge explosion of potential combinations of R groups, what we call a combinatorial explosion. So you might want to consider a bit of restriction on some of these rules that you can do. Now, one of the other transformations we talked about is a homologation. A homologation is an insertion of a CH2 or a methylene unit between heavy atoms. This creates the famous series of analogs, methyl, ethyl, propyl, butyl, butyl, and so on. They are generally inserted in chain bonds, but they can also be inserted in rings as special cases. There is homologation of heteroatom to carbon. So for instance, if you look here in the middle of the slide, you see you have a methylene unit and then a heteroatom. You can imagine that if you can create this molecule, it's probably synthetically reasonable to include an extra carbon unit, and you can also synthesize that group. In the same way, a pi carbon homologation is also often reasonable to do. So if you have, say, a CH2 unit and then a keto, you might very well be able to insert an extra CH2 unit. Again, as before with the bioisosters, we have to think about the framework of the rules, that there might be things we want to avoid. So for instance, we might want to avoid methylation on hydroxyl or thiols, for instance, on sugars, because a change of a hydroxyl group to a methoxy group on a sugar could really change the properties of it. We also talked about heterocyclic substitution. So here in the middle of the slide, we have a benzyl ring. Now, a benzyl ring is very similar to, for instance, a thiophene, similar size, similar polarity, hydrophobicity, also similar to pyridine, and also similar to the indole ring, and even also to this oxime. Again, that's one of those that is a bit surprising, but nonetheless. Now, what you will notice on this slide is that several of these heterocycles are interconnected. So you can imagine if you start with a phenyl ring and say you go to a thiophene, you could actually transform that thiophene once more to, say, a furan or to maybe a pyrrole. So we can say that this thiophene is a bridging transformation that leads us to even more diverse compounds. So if we allow the compound to change not one step, but two steps, or we call this iterations, we can greatly enhance the diversity of compounds that we can create. So how do we specify these transformation rules? Well, we have chosen to use a standard format, so an RxN format, because that's available in standard chemical sketches, such as ChemDraw. The real benefit of that is it's easy to specify them and visualize them, much easier than, for instance, using smirks. The way it's written is like a reaction. So imagine you have your thiazole that you will say you can be transformed to an ester. You'll write thiazole, then an arrow, and then you'll have your ester. Then when you translate that into an RxN file, you have the same chemical entities, but you can use the extra formats in the RxN file to be more or less specific. So for instance, position number 4 here could match a sulfur, as we talked about, a thiazole, but maybe you also want to match an oxazole. So you could say oxygen, comma, sulfur, and then that reaction would match either of those two heterocyclic rings. Now, what we also have is some numbers added on the heavy atoms. So for instance, imagine our thiazole in the input, it has an R group. Where does that R group go? Does it go on the carbonyl side of the ester, or does it go on the ether side of the ester? Well, with the reaction file with the numbers, we can see that that R group belongs to number 1, and number 1 is transformed over to the carbonyl side of the ester. So we can use these numbers to indicate exactly how they are transformed. Now, if you have many similar transformations, you can have a directory, or you can have them in a database as we have in Mo, or we can even have subsets and classifications. So you could go in and choose, let's say, all the transformations that goes from an ester to an N-based heterocyclic rule and say, those are the transformations I want to use. Now, again, there are considerations that has to be done if you were to go and create these transformations yourself. For instance, how do I deal with changes to aromaticity, formal charges, and substituent counts? Because again, fundamentally, we want to create new compounds that are not too dissimilar from our original compounds. So once we have the reactions, how does the transformation algorithm work? Well, basically, it's a match and replace. So we have our transformation, and if our input molecule matches the left-hand side of the transformation rule, we delete those atoms and we replace them with the right-hand side of the transformation rule. If there's any ambiguity, we have the atom numbering to help us out. If there is a way of matching it multiple times, we will do all the changes. So here's an example. Imagine you have the transformation going from a piperazine ring to a pyrrolidine ring, like you see here in the box to the left. Then you have your input molecule over to the right, and that indeed has a piperazine ring. Now, because of symmetry, we can actually match a piperazine ring into that input molecules in 4 different orientations. However, there is a restriction on the transformation saying carbon number 3 can only have 2 heavy atom substituents. So that parenthesis S2 means 2 heavy atom substituents. Because our input molecule has one of the carbons with 3 heavy atoms on, that will not be able to match as carbon number 3. But carbon A, B, and C could all be interpreted as carbon number 3, and then the rotation of the rest of the match would go around the ring accordingly. And that leaves us with being able to create 3 different compounds, A, B, and C, as you see down at the bottom of the slide. Now, it turns out that compounds C and B are quite hard to make synthetically, whereas A is easier to make. And because you can see we can quite quickly generate many different molecules, just a small molecule like that, just one transformation gives us out 3 compounds. You can see how that can become a lot of compounds all of a sudden. We do want a way of saying, actually, we are only interested in those that are actually synthetically feasible to create. Basically, we do need a way of looking at the synthetic feasibility, because several transformations can actually generate molecules that we just can't make. Like a transformation, even though it looks like a reaction with an arrow in, it is not a reaction, right? And changes to the carbon skeleton can actually be really hard to make. So we went about and we wanted to create a score that we call synthetic feasibility, or RSynth. The way we did was we took the new suggested molecule, so the modified molecule, and we fragmented that molecule based on an extensive set of rule-based retrosynthetic disconnections. In another word, if you got this molecule as a chemist, how would you synthesize it? Well, you would break it down into fragments that you knew you could stick together to the full molecule. So here we have our molecule, it's been fragmented, and here is the fragments that make up the input of that molecule. Then we look up each of these fragments in a big database of starting materials, and we see how many of these fragments can we find in that database. If we can find all of them, we know that it should be possible to create that molecule based on the standard ways of creating molecules for different functional groups. And we give it a score that ranges between 0 and 1. So it's been normalized to 0 and 1. And 0 indicates, we think that will be hard for you. And 1 indicates, we found all the fragments. We think you should be able to create this molecule. So we can combine this RSIN score together with other descriptors for filtering and ranking of all the output molecules that we can come up with. Now, another important consideration is not only do we want to make synthetically feasible compounds, but we also want to have compounds that are similar in 3D shape to our input molecule, right? Because fundamentally, they have to fit into a binding pocket in 3D shape. So we use something called 3D embedding to ensure we have similar shapes of molecules. The way we do it is we identify the parts of the molecules that have not changed, and then we try to keep their relative distance unchanged when we do the transform. As part of that, we allow the active site of the protein to be part of it as well, of course, to ensure that the molecule will still fit into the pocket. There are easy cases to do 3D embedding on. So here in the middle of the slide, you see one of the transformations we looked at before, amide to a vinyl fluorone. Again, they were very similar. They were similar size. They had the similar exit vectors out. That's an easy case of replacing. If we have changes to the terminal ends of the molecule, that's also relatively easy to deal with. However, for the hard cases, such as the one you see down at the bottom, where we have insertions or deletions of atoms, particular in rings, we do really not only change the size, but we also change the relative orientation of that molecule. So imagine the R groups in the 6-membered case here, they suddenly get a bit closer when we have a 7-membered ring after our transformation. So in cases where it's difficult, we create multiple attempts of 3D shapes to find the one that's closest to our input molecule, and we also pick the lowest energy conformation out of those many tries that we have done. So I want to show you a couple of examples of how embedding works. So imagine you have Gleevec, the molecule Gleevec up in the top left corner, and we are generating a couple of analogs. We start with a homologation. So the first step here has added a methylene unit before the amide group. That's a bit of a tricky one, right? We have added an extra carbon in the middle of the molecule and it suddenly gets a bit longer. The other two analogs we have here are changes from phenyl rings to furan rings on either side of the amide group. Again, it's a reasonable heterocyclic substitution, but it is a bit more tricky because it's in the middle of the molecule. They have a bit different size, and they also have a bit of different ways of connecting with the angles to the rest of the molecule. Nonetheless, when we do 3D embedding, you'll see the results up here in the top right corner of our molecule of the slide. They actually, all three of them here are superposed on Gleevec, and you'll notice they have a very similar size and shape, all of them, right? So we can actually make them contain within the same shape as before. And even, for instance, the location of the amide here is in the same area. So if it were to interact with the protein there, it would still be able to pick up the same kind of interactions. Now, down on the bottom corner of the slides, we have allowed Gleevec to have even more analogs. So 132, we've embedded them, and you still recognize the shape of Gleevec in there, right? You can still see that same shape and about the same size. But as we generate more and more diverse analogs, some of them will struggle to maintain the same 3D shape. So for instance, you'll notice that there's an indole ring here that is sort of protruding. And if that were to clash with the protein, it might not fit. So say we were to include the protein as well in this calculation, we could very well have to exclude a lot of these molecules because they just wouldn't fit in the pocket. So one thing is, of course, looking at the shape. Another thing that's quite important is looking at the properties of the compounds as we start to modify them. So here we've analyzed Gleevec again, and we allow Gleevec to change in 3 steps, so 3 iterations. The green color is the properties of the molecules after 1 iteration. Then we modify those molecules once more, and the properties are shown in iteration 2 with red, and then iteration 3 in blue. If we start in the top left corner, we see the molecular weight of each of these analogs. And you'll notice that it does increase ever so slightly as we go over each of the iterations, which is entirely understandable. For instance, when we do homologations, we actually insert an extra methylene unit, right? But nonetheless, they do still stay within a relatively reasonable area of molecular weight compared to the input molecule. Down below, we have a histogram of number of rotatable bonds, and we can see that it does increase a bit. So we have fewer rotatable bonds at iteration 1 than we do at the higher iterations. Again, it's an indication of how we've diversified the compounds. Now, interestingly, though, if you focus over on the right-hand side of the slide, we have a couple of properties that will say something about the polarity and maybe the bioavailability of these compounds. So at the top, we have log P, and below we have TPSA, which is the polar surface area. And here you'll notice how similar these properties stay despite us generating more and more diverse compounds, because basically we want to still maintain compounds that are similar in properties to our input molecule. So now that we looked at each of the steps, we can summarize the methodology that we use. We start by taking our input molecule, imagine that's the one up here at the top, and we take all our transformation rules that we want to apply and apply them. So imagine the first transformation rule says, if I have a hydroxy, make it into methoxy. We look, oh, I have a hydroxy, let me replace it with a methoxy. Then the next rule says, if I have an amide, replace it with a vinyl fluoro. Oh, I do, so let me replace it, and so on. That generates, here in this case, 4 molecules. We're going to embed them in 3D and refine their coordinates. Then if we allow multiple transformations, we take each of these 4 molecules and we run the same rules on them once more. You might notice that you can get the same molecules. So say you do transformation 1 and then transformation 2, it will give you the same molecule as if you did transformation 2 and then transformation 1. But of course, you will eliminate those duplicates for you. You don't have to worry about looking at the same compounds multiple times. And at the same time, we will, of course, use all our filters to limit the output so that we only get out reasonable molecules at the end of the calculation. So the last part I'm going to show you here is a case study. It is actually about sildenafil in the PDE5 pocket and how we can transform it. All right. So first, a bit of introduction to the PDE5. So the PDE5 is the enzyme that breaks down cyclic GMP. It's found in smooth muscles, so that's the corpus cavernosum, lungs, and retina. There are several marketed inhibitors of the PDE5, for instance, sildenafil and vardenafil that we looked at, but also tadalafil. I have found a crystal structure, a publicly available crystal structure of the PDE5 in complex with sildenafil, and you see it here in the top right corner. We have the protein in gray, and we have sildenafil in its binding pocket in green in the middle. I've then chosen to zoom in on the binding pocket so we can have a look at how sildenafil interacts with the protein. So if you focus down in the bottom left corner here, we have in the middle of the picture in line drawing, we have sildenafil. And around sildenafil, we have the nearby residues. They are color coded so that if they are pink, or let's call it purple pink, they're polar. And if they're green, they're greasy. On each of these residues, there is annotated if they interact with a specific interaction with the ligand. So for instance, in the scaffold of the ligand, in that fused ring scaffold, we have a cyclic amide. And that cyclic amide is very important for binding because it makes 2 hydrogen bonds up to glutamine 817. So the amine of the scaffold interacts with the carbonyl of the side chain of glutamine, and the carbonyl of the scaffold interacts with the NH of the side chain of the glutamine. And that creates 2 really strong hydrogen bonds up to that glutamine side chain that really is important for that binding here. Now, the rest of that scaffold has several arene interactions. So there is a pi-pi stacking from the ring and up to phenylalanine 820. And there's a proton pi interaction from that scaffold and to valine 782. Now, furthermore, there are several strong surface contacts placed around. So for instance, we have phenylalanine 782, we have a leucine 84 and leucine 765 that are all important for interacting with the ligand. You'll see that there's also several bridging water molecules that are creating hydrogen bonds from the ligand and then bridging to different residues in the protein. What we also have is an indication of the pocket of the protein. So the dotted gray line that you see around the ligand, that's the wall of the protein. It indicates how well this ligand sits in the pocket. Now, what you might notice is around where my cursor is over to the left, that dotted gray line is broken. So that indicates that the pocket is more open there. We generally say that's the mouth of the pocket, so the opening of the pocket. And you can also see that the ligand atoms on the left-hand side of the molecule have these blue clouds on, indicating that they're solvent exposed. So that would be where the ligand comes in, where the pocket is open. And then the right-hand side of this drawing is then the deeper end of the pocket. Now, talking about the pocket, that really is a better thing to do in 3D. So I switched to here a snapshot I took of the 3D view. So here again, it's still sildenafil. Sildenafil is colored green, and it sits in the pocket. You can see the nearby residues around it. Now, I've highlighted this really important glutamine residue in what we call a ball and stick representation here to show you the hydrogen bonds as these dotted blue lines with cylinders on it, going from the amide group in the side chain and to the amide group of the scaffold, right? Now, when we start transforming sildenafil, these hydrogen bonds are very important, and I want to maintain at least one of them in my transformed molecules. And the way that we do that when we run molecular transformations is that we add something called a pharmacophore. And you'll see that I've added in two features in my pharmacophore. I've added in one sphere around the carbonyl oxygen, which has the type of acceptor. What that tells the program is I want to find a hydrogen bond acceptor within that sphere. But I added an additional feature as well, saying not only do I want to find an acceptor there, but I want that acceptor feature to point in the right direction. So the larger sphere is what we call a projected acceptor point, and it gives the directionality of that lone pair on the carbonyl, making sure that it actually points up towards the NH to interact with, and not, let's say, completely different downwards, because then we wouldn't create the hydrogen bond anyway. I have also indicated the binding affinity that has been scored with our generalized boron implicit solvent scoring function, which we also use for our docking score. And we have around minus 9.9 kcals per mole. And I have added in the force field interaction, so around minus 65 kcals per mole, and of course also the arsen score of sildenafil of around 0.64. Now, the final thing to point out before I move to the next slide is the shape of the pocket. So the gray mesh you see is our interaction surface. And the interaction surface indicates where your ligand atoms should be placed. So you should focus on placing your ligand heavy atoms on this mesh, because when they are in the mesh, it indicates that they are in a good distance to make interactions with the protein. You'll actually see that exactly with the amide, the nitrogen, and the carbonyl oxygen. They sit right on that surface, as well does the nitrogen, the pyrazole ring. Now, what you might also see, and I'll rotate out the next snapshot, I'll rotate a little bit is, but what you might see already here is that the three R groups, so the propyl, the ethoxy, and the piperazine ring, they actually sit, in a pocket that is larger than the space that they take up. So let's just rotate that view. This is the rotated view, right? You see now from the mouth of the pocket, and you see the piperazine ring here in the front. You'll notice the ethoxy ring. It doesn't really touch the surface down here. There is room to do modifications to increase the fit between the ligand and that pocket. The same goes for around the propyl chain here in the back, and of course where the piperazine ring is in the front. But interestingly, when you look at the very, very deep end of the pocket, you go and look in there, you'll see how narrow that pocket is. So right around that scaffold, that pocket is really narrow. There's not a lot of room to deal with there. And we have to think about that when we do molecular transformations. Now, that is one of the real benefits of doing 3D embedding with refinement in the pocket, right? That all the molecules that we get out will be ensured to be able to fit into that really narrow pocket as our current molecule does. If we were just going to do that in a 2D sketch, it would be really hard to know which of them would actually be able to sit like that. It would be interesting, now that we do have sildenafil, to actually make the transform to vardenafil. And just to recap the kind of transformations that is needed, right? We need two steps. We need an NC switch to match on the pyrazole ring to make it an imidazole ring. And that would be the reaction file you see here in the box over in the top right corner of the box. And then we need a homologation that will take the methyl group on the piperazine ring and turn it into an ethyl group. And that will be that reaction file with the heteroatoms that I showed you before. So one methylene unit, then I can insert another methylene unit. So all in all, if we want to reach vardenafil, we need to allow what we call two iterations. So to do one step and then the other step. So the calculation we set up was as follows. We took our 1 TBF complex and we ran our preparation protocol quick prep to prepare for missing atoms, set protonation states, set the tethers and restraints so we can do a refinement of the overall structure to relieve it for any clashes and strains. Then we applied our transformation rules in 2 iterations so we could reach vardenafil. And we then used different 2D filters and our 3D pharmacophore. We refined the molecule in the active site. And finally, we scored it with our generalized boron implicit solvent scoring function with a weighted term for exposed surface area. And of course, at the last step, we also included synthetic feasibility. And the results are, as you see at the bottom of the slide here in the table. If you start with the first row, I did 1 iteration. And out of 1 sildenafil molecule, I could generate 87 compounds. Then I applied different filters. And the footnote here will tell you that it's a molecular weight less than 550, log P between -4 and 8, and a polar surface area between 40 and 140 square angstrom. So standard filters really for bioavailable, orally available small drugs. Now, applying just these filters actually reduced 30% down. I'm down to 61 compounds then. We then had embedding and refinement in the pocket, and actually the same 61 compounds remained, again, because we haven't changed that molecule very much. Applying the pharmacophore query just threw 1 molecule out, again, really underlining how we haven't changed that molecule that fundamentally much. And finally, we applied the RSynth score. And here we said, I'm only keeping those whose RSynth score is above 0.6. And that led me down to 40 molecules, so more than a half or more than 50% reduction with my filters. The final number is an RMSD of the unchanging parts of the molecule. So the parts that wasn't transformed, how much did they have to move when I transformed it? And you'll notice that that number is really low, between 0.1 to 0.4 for these molecules. So they hardly had to move, again, indicating that that molecule sits in the same way in the pocket as it started out with. Then after that, we went along and we said, okay, let's do a second iteration. And if we just do all the molecules we can achieve from 2 steps away from sildenafil, that's a whopping 3,663 molecules. So that's quite a lot, really. We started filtering then. First, we did the descriptor filters and we were down to 3,200. Then we included 3D embedding and pocket refinement, and we took another half off them, so another 50% off. Again, now we're starting to generate more diverse compounds, right? And they're a bit further away from sildenafil, and some of them just won't fit. So they were filtered away as well without us even having to look at it. We then implemented the pharmacophore filter as well, and we got away with another 100 or so compounds. And finally, we added in the RSynth score, and we were down to 670 compounds, so almost a 6th of what we started out with. So a really big reduction in the number that you actually have to look at from the starting potential number of compounds. And again, of course, we looked at the RMSD, and it again stayed very similar to our input molecule. So I have a couple of snapshots from that. The first one here is after the first iteration and after the 2D filters, here's what the molecules look like in the pocket. So these are the 61 molecules. I only embedded them, but I didn't refine them. And you'll see what I mean with very similar shape. They sit very similar in the pocket, very similar binding mode, right? When you then refine them, a few of them will move a bit around, maybe because there's a mismatch in the electrostatics, maybe there is some steric thing that doesn't fit exactly well, and some of them will move a bit, and if they move a bit too far away and they don't fit, they will of course be removed, or they will score badly in the affinity score. Here, I've shown you that we did generate vardenafil. So in green, we have sildenafil, and in purple, we have vardenafil. Look how similar they sit in the pocket, right? They have the scaffold is exactly the same place. They have the same interactions to glutamine. The only thing that's changed is that we have that pyrosyl ring that's now an imidazole ring, and we have a homologation filling out that lower pocket a bit better. We have a slight increase in affinity, so minus 0.1 kcal per mole, and a very similar RSynth. So at this slide here, I'm showing you a couple of the compounds to just give you a bit of the diversity of compounds that we generate. I first of all want you to notice that the majority of transformations actually happens out in the R groups. So not so much on the scaffolds, right? And that's because we know that that scaffold sits really well and there's limited changes we can accommodate. But we do know that the R groups didn't fill the pockets very well. So it's not odd for us that we do have some optimizations going on the R groups. So let's look at the first one here. We have a homologation creating a longer ether group there. And we also have a replacement of the pyrazine ring with a pyrrolidine ring. We also, at rank number 4, we find an NC switch like the one we had in vardenafil, and again, a homologation of the same R group. We see a couple of changes to that R group. There's one of them where the ether is replaced by a keto over 2 steps. We also see some optimizations to the R group of the pyrazole ring, for instance, here with a triple bond instead. And if we go a bit further down, we do actually see an update to a scaffold here where the amide has been replaced with an ester. That's actually perfectly reasonable, and I have a 3D shot of it, which I'll show you in just a minute. So here is the best ranking one in after our filtering and scoring. Again, sildenafil in green, the new compound in purple. It had the homologation that increased the ether group up at the top of the phenyl ring, and you can see how it fits much nicer in the pocket up there now. And down where we had the pyrazine ring, we now have pyrrolidine ring that also fills the pocket nicely. And again, we have a slight increase in affinity as compared to sildenafil, and we still have a very similar RSINT score. Here's one of the other ones. Again, the homologation up in that subpocket, and the NC switch again sits really nicely, as we also know because vardenafil sits really nicely. Slight increase in affinity again, very similar RSINT score. Here is the one where we have a change to the scaffold where the amide has been replaced by an ester. Now, what you will notice is that replacing that amide by an ester, it actually loses one hydrogen bond, right? So that amide hydrogen there no longer is there, and we don't have one of the hydrogen bonds to the glutamine. But nonetheless, we are still able to get a nice affinity because we still have that strong hydrogen bond between the carbonyl group and the side chain that we had encoded with the pharmacophore. And we also gain extra affinity by homologation on the R group on the pyrazole ring that now fits that subpocket really nicely. The last one I added in here is actually the one that scores the worst in our suggestions of compounds after the filtering and ranking. So a couple of things happened. So there's been an ethyl to an ether change on the R group, but really interestingly, what has happened is a change to our scaffold, right? So the amide there has been replaced by a hydroxyl ethyl. That is still okay within the pharmacophore in that we still have a hydrogen bond acceptor in that location. Now it is a much weaker acceptor. So those hydrogen bonds are much weaker than before. And also we have lost that same hydrogen bond with the nitrogen amide that is also not there. So those changes are 2 large changes away from our original compound. And we can see that by it ranking really poorly. So as I said, it's the last one. And also we have a higher increase in affinity. So almost plus 2 kcals compared to the original molecule. And keep in mind, we want those numbers to be as negative as possible. So we don't want to add any numbers to it. Just to finish up, really, the MedChem transformations are easy to use. They're integrated with all our other fragment technologies. So we have, for instance, link separate fragments. We can grow R groups on our ligands. We can do scaffold replacements, and we can breed, which is a hybridizations of crossing over ligands. So kind of like if you have superposed ligand bonds, you can create crossovers of the 2 of them. Now, because they are all within the same framework, we have the same protocol to deal with them. That includes our 2D filters, so that's more than 400 descriptor filters, QSAR models, fingerprint models, if you have that as well. Pharmacophore filters in terms of 3D filtering, energy minimizations with a very flexible receptor, and 3D scoring, so for instance, our docking score or an RMSD movement of the score, and of course, our synthetic feasibility score as well. So we have our unified fragment panel for all the different fragment tools. I've showed it here for the MedChem transformations. In there, we have a reaction file with 175 default reactions that you can, of course, create custom versions of. You can apply the 2D and 3D filters. And you can include all the same filters in that panel at once. And you can also in the same panel choose how you want to refine the ligand in the pocket, if you want to do it as a rigid receptor or as a flexible receptor. So that really leads me to the end. And I think it's over to you, Val, to see if we have any questions.
Thank you for the great presentation, Sarah. Yes, we've actually had many questions. I don't think we'll go through all of them right now, but maybe I can ask a few of the questions that we did get. One of them is, can I specify which transformations I want to take place?
Oh, yeah, that's a good question. So I'm not exactly sure what you mean. You could interpret that in a couple of ways. So if you don't mind, I'll show you what I'm thinking of in Mo. So maybe I'll just hide the slideshow there so I can open my Mo window. There we go. I have Mo here. I actually do have the same system already prepared. So I think I'll use this as an example. So I have here one TBF that we have prepared with quick prep. And again, you should recognize this view, right? We have the protein around in gray, and we have sildenafil in the middle here in green. And because it's already been prepared, I can show you a couple of transformations on this. So if you were to run molecular transformations, you would go to the header to compute fragments, and here you have a list of all those fragment tools, including medicinal chemistry transformations. So here you have the unified panel for all the transformations, and it's split into those 4 sections that I mentioned. Up at the top, you define the ligand and the receptor. In the middle, you have the settings for the specific fragment tools that you want to use. The third part is for filtering, so descriptors, model files, pharmacophores, and the bottom part is for minimizing and scoring. Now, what you might notice here in the second part, so the part with the settings for the medicinal chemistry transformations, is it in green says no atoms selected. So if you were just to go ahead and press search down at the bottom of this panel, it will transform on the whole molecule, so the whole sildenafil. But you can actually specify parts of it that you can transform on. So let me just move that panel out and open the 2D view on the footer and say, I just want to transform, let's say on the sulfone and piperazine ring like this. When I select atoms on the ligand, that panel automatically updates and says, you have selected 22 atoms, and that indicates I'm only going to transform on those 22 atoms. So that might be one way that you can specify it. Now, the other way that I'm thinking of you might find useful is to actually specify the specific reactions you want to do. So if you look here in the panel where it says transformations, there is a reference to a database, and that's the database that holds all of those 175 standard reactions we have. And we can open it with the database viewer right from here. And when you have that database, I generally myself, I prefer to actually browse them. So if I go to File and Browse, it will show me the exact same view as if you loaded that reaction into a sketcher, right? And let me just switch to the third one here, because we should recognize this one. If you have a sulfonyl group, you can replace it with the group on the right here, an ether. So if you want to do that transform, you can simply toggle on select. You could look through them and say, okay, actually, I want that one too. And you can select that one too. And you could go through and select the ones you find interesting. Alternatively, if you don't want to look through them, you can, of course, also select them based on their name, because they're actually given a name for their functional groups. So you can always right-click on the header, say select, and you can write, let's say, we need to put it in single quotation marks. Say you're interested in amide transformations, right? You click enter, and it will select all the reactions that have amide in the name. And if we just go ahead and we hide the unselected ones, you should see the reactions here. And if there's some of them you don't want to use, maybe some of these retro ones, you can simply toggle them off, right? Go back into the panel and toggle on selected transformations only, and it will only run that subset of transformation. Again, as I mentioned, you can of course have them in a separate database or a separate folder as well. Now, the final thing, and that's just because I'm not exactly sure how you meant with specifying the transformation. I want to show you just one more way of doing that. So let me just tidy a bit up here. I'm closing the database of reactions and just toggling off that one again. Now, say you want to transform sildenafil, right? And you want to transform the whole molecule, but you want to specify that the phenyl ring should be a pyridine ring. You don't really care about the rest of it. You just want a pyridine ring where the phenyl ring is. How can we do that? Well, that's with a pharmacophore. So the pharmacophore you can actually create on the fly straight from this panel. If you go down to the section of filters, find pharmacophores, you can open the editor. As soon as you open the pharmacophore editor, you get these so-called annotation points, these colored dots that indicates to you where Mo finds important atoms or groups of atoms that you can choose to make features out of, exactly how I made a hydrogen bond feature from our glutamine. Now, the pharmacophore editor itself is set up in the typical Mo sections, of 3 sections. Up at the top, there's settings for the annotation points. In the middle, you'll build up your list of features. And here also, and that's the key to this one, you can modify them. And down at the bottom, you can save and search. So how would I go about making a pharmacophore that requires a pyridine on my phenylase? Well, let me move that out of the way again. And I'm going to take advantage of the center here of the ring. That's what we call an aromatic feature. So I select it and I make a feature out of it. And right now it says, okay, this is a feature that's aromatic. And if I leave it like that, I will require to have some sort of aromatic ring there. But I want to go one step further, right? I want to require a pyridine ring. But that feature is completely customizable. You can use Boolean expressions, you can use and, or not, or you can put strings on them for that matter, but you can also put smart patterns in here. So say I'm just going to delete that one there. And I need to start with double quotation marks here. And I'm going to count 1, 2, 3, 4, 5 carbons, 1 nitrogen. I've now said within that sphere, it's not aromatic anymore. I want to find a 6-membered aromatic ring with 1 nitrogen in. I might want to increase the size a bit just to be a bit more lenient like that. And then when I go back to my panel, maybe I just want to allow 2 transformations in this one and just start it otherwise with default. So yeah, I haven't added any refinement or any scoring in here. What you should be able to see is that I create output molecules in the first column. The second column tells me the rule that I've been using. And you can see the first rule used is benzene to pyridine. Then for the molecule 4 and downwards, that's actually my iteration 2 I've generated. You can see the column here, number transformation, it's from 1 to 2. And now I'm modifying one step further away from the benzene to pyridine so I can transform the rest of the molecule. So I'm not exactly sure if that answers your question. It was 3 different ways of doing that. If it wasn't the right thing, please just send us an email. We can, of course, have a call about it. In the meantime, let me just cancel this one here to see if there is time for more questions, Val.
Yes, so one question that came up several times actually is when you do the refinement in the pocket, is the protein allowed to move?
That is actually a really good question. We didn't talk much about it, but let me show you. So again, here in the panel, you will need to toggle on minimize to refine it in the pocket, right? And when you toggle that one on, you have a settings wheel. So this gear wheel is the Mo settings wheel. And when you click on it, you get settings for the refinement. And what I wanted to point out to you here is by default, the side chains are fixed. So that means the protein is rigid while you allow the ligand to move around in the refinement. Now, if you want to allow the protein to move a bit, you can change that. So for instance, you can tether them with a certain strength so they can move a bit, but not too much. You can tether them by their B factor. You can set them completely free and they can move, or you can use the current tethers you have. So these are the different options where you can allow a bit more movement in your protein.
Perfect. Maybe one last question. Is there a way to quickly generate analogs either in 2D or without the receptor?
Yeah, sure. Actually, we didn't talk much about that either, exactly because we talked a lot about these 3D settings. But yes, of course. So let me just tidy up a little bit here, close the pharmacophore editor. We don't need to save it this time. So depending on exactly what you want to do, there's a couple of options. Option number 1, if you just want to embed them in 3D, but you don't want to include the receptor, you go to the top of the panel and you switch receptor to none. If you run it like this, you will still do the 3D embedding to make sure the ligand is in the same shape, but you won't consider the receptor as part of that. Alternatively, if you actually just want 2D molecules out, we have the settings for the transformation. And in the pop-up here, you can see generate 2D. And generate 2D, if you toggle that on and click OK, will only generate you 2D molecules, which is, of course, a really fast calculation if you just want to get out all possible combinations that you can do.
Perfect. Thank you very much, Sarah. That was a great presentation. I think that's all the time we have for today. So I'd like to thank you, and I'd like to thank everyone for joining us today. If you have any additional questions or ideas for future webinars, please don't hesitate to email us at the address you see on the screen right now. So that's support@chemcom.com. As I mentioned earlier, within the next 7 days, you will receive an email notification with details on how to access the video recording of this webinar. So thank you again and have a nice day.
Thank you for today. Take care.